Hi, my name is Christine Byrne, and I've chosen to do prompt 1 for exam video 5, discussing the adapter hypothesis developed by Francis Crick. As an overview, the adapter hypothesis was used to describe the function of tRNA molecules during the process of translation in cells. Essentially, Crick proposed that tRNA acted as an adapter or connecting device used to carry amino acids to their correct location on a transcribed mRNA strand. Take this as an analogy. If you're interested in listening to music on this phone but only have these headphones, no sound will be produced through these earbuds because this cannot be directly plugged into this source of sound. But if you have this adapter, one end is compatible with the phone and the other is compatible with the headphones. Thus, sound can now flow through these headphones. Similarly, when tRNA acts as an adapter, one end gives way for base pairing to connect the tRNA to the mRNA, while the other end carries the correlating amino acid that can then be incorporated into a growing polypeptide chain. Let's get a bit more specific. Once transcription is complete, there is an mRNA strand that holds a code for the creation of specific proteins. In other words, it's ready to be translated. This process takes place in a ribosome, a 2-subunit complex made of proteins and rRNA. But the really important part is the tRNA molecule. tRNA stands for transfer RNA and is a sort of T-shaped molecule made of RNA nucleotides. It's the physical link between mRNA and the building blocks of proteins. The structure of tRNA is such that it contains an anti-codon loop that allows base pairing to occur between it and specific codons on the mRNA strand. The 3' end of the tRNA contains a terminal site for attaching the amino acid that corresponds to the codon that specific molecule attaches to. The process of translation begins when the small subunit of the ribosome binds to the mRNA strand along with the tRNA molecule whose anticodon recognizes and pairs with the start codon AUG. This codon corresponds to the amino acid methionine. Once paired, the large subunit of the ribosome then binds, establishing 3 translation sites within itself. Moving in the 5' to 3' direction, the next tRNA molecule then pairs with the codon following the start codon, bringing with it its attached amino acid. With the first tRNA in the P site and the second in the A side of the ribosome, the 2 amino acids link, forming the beginning of the polypeptide chain. As the ribosome moves along the mRNA strand, each previous tRNA molecule moves through the ribosomal sites from A to P to E, where it has successfully dropped off its amino acid and is ready to exit the ribosome to go find its next amino acid. In sum, tRNA acts as an adapter for 2 main reasons. 1, it contains an anticodon that allows it to pair with the mRNA strand, and 2, it carries a corresponding amino acid at its 3' end that allows the creation of proteins. Knowing this, an alteration at its anticodon loop could cause a tRNA to not follow the universal genetic code. For example, if a tRNA is attached to the methionine start codon via an amino-acyl tRNA synthetase enzyme, but undergoes a mutation causing its anticodon to read GAC instead of UAC, then base pairing between the tRNA and mRNA won't occur, and translation into a protein of this gene won't occur. This kind of alteration could cause cell death if the protein was vital for function.